organizing this. Here we got it, yes. Right, so without further ado, because I think it has been embarrassing enough, I'm just gonna jump into the presentation and hope that the screen sharing works as beautifully as during our trials. If not, scream and let me know. Is that all right? Yeah, I see nodding. Brilliant. Okay, right. So as Julia mentioned, we kind of have quite a diverse set of topics that we're trying to cover in the lab and frequently using museum collections. And today I decided to tell you about some aspects of our work on dental calculus, which allows us to travel through time using metagenomic data. So I will try to cover two topics, but if I run out of time, you'll please have to scream. So then I'll stop halfway through. But I want to tell you the stories about how their oral microbiome can tell us a story about use of antibiotics by humans and about consequences of population decline. And before I start and talk about science, I want to talk about the people who actually carried out this work, because as group leaders, we're frequently the ones who worry about the financial aspects and the logistic aspects, but a lot of science and most of the science is being carried out by our group members. And in this particular case, Jayla Brealey, who was a postdoc in my lab for some years, has been absolutely driving this work forward. And I'm extremely proud of her and extremely proud of the work that she has done. Adrian Forsyth is a current postdoc in the group, and he is primarily driving forward the work on their oral health. And we had a number of fantastic master and PhD students and trainees that have contributed to these projects throughout the year. So they all deserve, recognition for what has been done and what is in the process of being done. Right. So this entire topic is about host-associated microbiome. So these are collections of microorganisms that live inside and outside on the host. And for instance, you can think about the gut microbiome or the skin microbiome. And what separates the host-associated microbiome from the free-living one is that it is impacted by two major kind of groups of environments or processes. So they're the extrinsic environments where the host lives. So for instance, it would be aspects such as temperature, humidity, maybe geographic location, but they're also the intrinsic host environments. And these are primarily host genetic factors and immune factors that will control what type of microorganisms are allowed to colonize the host in the first place. So there are these two mechanisms that structure these meta-communities that live on the host. And in turn, of course, the microbiome itself impacts the host in very prominent ways. And there's a magnitude of biological processes that the host microbiome has an influence on. If we think about the gut microbiome, digestion is the obvious one. But microbiomes, host-associated microbiomes, have also been implicated into adaptations to changing environments, particularly important now, behavior and reproduction, and many other aspects. And of course, lots of diseases. So it is an important aspect, important enough that there's whole groups that do hologenomics. So basically trying to understand this complexity of macro and microorganisms as one evolutionary unit. However, these host-associated microbiome evolutionary processes have been kind of under-investigated, in my opinion. And the reason for that is that we tend to study them on two very, very distinct and very contradictory timescales. So either we look at these processes over millions of years by comparing microbiomes from different host species. For instance, we can look at the gut microbiome from a set of primate species, as shown here from a paper published not too long ago. And there we can see that the host microbiome can be actually clustered and structured, and the structure and the similarity or dissimilarity of the gut microbiome of different primate species actually corresponds quite well to the primate phylogeny, suggesting that there's a co-diversification, if not co-evolution, of the gut microbiome with the host species. But these are processes that took place over millions of years. On the other extreme end, we know that host-associated microbiomes can respond very, very rapidly to stimuli, such as, for instance, changes in diet or changes in the reproductive state. And these responses can be as rapid as following months, weeks, days, or even hours. So we can see changes in the microbial communities within an hour of a stimulus. What is lacking, in my opinion, to make this claim of co-evolution of host microbiomes and their hosts are these intermediate timescales that span millennia, centuries, or decades, where response to the environment or a demographic event within the host could be seen by studying the microbiomes. So what type of processes I'm thinking of? Here's an example: climate change. Particularly relevant, we are finding ourselves in a situation of extremely rapid climate change. We do not know how this climate change is going to impact host-associated microbiomes and what type of consequences it will have for the host. But we could study this process if we go back into the past. Here's an image of the macrofauna of the last ice age. Of course, a lot of these large mammalian species went extinct, but the white arrows show a few species that actually survive the transition from the last ice age to today. There's a musk ox, there's an Arctic fox, which is difficult to see reindeer. So if we could study the microbiome of these survivors throughout the transition from the last ice age to today, we could understand what type of impact climate change has on host-associated microbiomes. Another interesting aspect is demographic change. Again, extremely relevant. We know that a vast majority of populations of wild organisms are declining and going through bottlenecks. So this graph is taken from the Global Living Planet Index, showing that populations of vertebrates are declining on average by about 70%, and this is over the time scale of the last 40 years or so. So how does demographic change impact host-associated microbiomes? We don't know. Or, if we think further, we can think about other levels of anthropogenic impact beyond population decline, such as contamination of the natural environments. Again, what type of processes, so what type of consequences would these events have? To understand that from the host-associated microbiome perspective, we actually need to travel back in time. So we need temporal samples and sampling over a long time scale. If you think about the last ice age, we need to go as far back as the last ice age. If you think about anthropogenic changes, we need to go back for at least 100 years or so. The problem is that microbiomes usually do not preserve after the host has died. So if we think about gut microbiome or skin microbiome, it's going to be degraded with the main body of the host. There is, however, one microbiome, well, actually two, but one in particular, that does survive this degradation or disappearance of the host. And this is a microbial fossil, and specifically dental calculus. So what is dental calculus? It is this microbial biofilm that builds on the teeth of mammals, and possibly even not just mammals, but other vertebrates as well. It is the stuff that we remove every day when we brush our teeth. If we don't brush our teeth, as has happened, for example, during human evolutionary history for quite a long time, this biofilm undergoes periodic calcification. And we still do not know what exactly triggers this calcification, but basically becomes a fossil on a living host. And then after this calcification is finished, a new layer of bacterial biofilm grows on top. And over the time period, it produces this solid deposit that you see in the middle figure, that is called dental calculus. So the middle figure shows dental calculus on a human tooth, but in fact, it turns out that this process is not unique to humans. It also happens in non-human mammals. And on the right-hand side, you see a picture of a reindeer where the dental calculus looks quite different, but it is still dental calculus. So this meta-community consists of bacteria primarily. So about 90% of DNA in this meta-community is bacterial DNA. And by studying this DNA, we can talk about the composition of microbial organisms, particularly bacteria, but also if we apply metagenomic techniques, we can look at their function. We can also study diet, and this is because dental calculus captures other biomolecules besides the bacteria. It captures saliva of the host, basically, so DNA fragments from the host. It captures remains of the diet. And this way, we can also study contaminants and pathogens in the host environment. So dental calculus in non-human mammals is actually quite widespread. And here's a small representation of different mammalian species where I have observed the dental calculus to happen. And I put it out here just to show the diversity of mammals and the diversity of evolutionary or ecological questions that can be answered with this material. So we have representatives from marsupials, so from pouched mammals and eutherians. We have carnivores and omnivores and herbivores. We have aquatic mammals and terrestrial mammals. We have different orders of mammals, and we can study a great variety of questions with that. And this is how dental calculus looks on them. So again, quite different from humans. And I think that is what has prevented the analysis of dental calculus in non-human mammals, just because it looks quite different. And today I'm going to focus on this upper row. So I'll talk a little bit about our work on dental calculus of these three species, because that's where we started from and where we started doing this type of work. And the first things first, when we saw this material and when we talked to our collaborators about this, the main reaction was, that's not dental calculus, that's something else. It looks so different from humans. We don't think it is what you think it is. So we started off collecting a bunch of samples and basically trying to understand what are these deposits. So we performed shotgun sequencing and decided, or tried to partition this community of microorganisms from this deposit into different sources. So this is an approach that tries to take this microbial community and, looking at its composition, tries to say which of the provided sources does it correspond to, which is most similar to it. So we decided to compare it to human plaque and calculus, shown in orange and yellow, because this is a microbial community that is really well described. And it's an oral community, that's what we're interested in. But we also included a bunch of contaminants, such as human skin microbiome, human gut microbiome, lab contaminants, salt from tundra. And by looking at our meta-communities, we were able to quantify how much of our community in this dental calculus corresponds to any of these sources. And indeed, you can see that for a number of samples, there's a very clear and high proportion of our sequenced data that is similar in composition to what is known to be human oral microbiome, which is very, very encouraging, which tells us that indeed we're working with an oral component. And of course, we have a varying degree of contaminants. And in particular, I want to point out, I hope you can see my mouse here, this reindeer from the last ice age. So they have been collected recently, but they date back to about 50,000 years ago. And they have this huge purple component that corresponds to tundra soil. So this is where the remains were found. So we do expect to have this level of environmental contamination. What is also quite interesting is that, proportionally speaking, the gorilla oral microbiome had the highest proportion of it being similar to the human oral microbiome. Is it true that gorillas are better preserved than, say, reindeer or bears? Or is it possible that we're just able to better describe what is present in gorillas because they are evolutionary closer to our source, to the humans? So we're working on this question right now, and I don't have an answer to this. But once we were confident that what we're working with actually has an oral component, so it is dental calculus, it does represent oral microbiome, we looked at whether or not we can actually distinguish our host species using these oral bacteria. And indeed we can. So in an ordination analysis, the host species cluster separately from each other. So there is a signature of host-specific composition and maybe even host-specific microbial species that help us distinguish the host. And I will show just a few more examples of things that we can do with this dental calculus to demonstrate the versatility of this material. So I mentioned that we can look at diet, and indeed we can quantify, or qualitatively assess the presence or absence of particular dietary items. Here's an example of dietary investigations into reindeer. We had two main groups of reindeer, one from Svalbard, which is collected really far north, so this is the furthest north distribution of reindeer, and another from mainland reindeer range. And for instance, we could detect the presence of willows, which is a typical reindeer diet, but also saxifraga, so a high Arctic plant that was only present in the Svalbard high Arctic reindeer. Not only did we detect plants, we could also detect eukaryotes, and in particular rumen eukaryotes, which also makes a lot of sense. Reindeer are ruminants, they regurgitate their food, so parts of their rumen microbiome will end up in their mouth, and that clearly can be incorporated in the dental calculus. So we can qualitatively assess their diet, but also, have conclusions about basically their biology. Moving on to bears and looking at the host DNA. So again, I mentioned we can recover a certain proportion of host reads from the dental calculus. And with these host reads, we can actually do quite interesting things. So for example, we were able to reconfirm or to trace the known history of colonization of Scandinavia by brown bears in particular. So here in the center, you see the map of Scandinavia and Sweden, here colored. And Sweden has been, or Scandinavia has been colonized by brown bears after the last ice age from two different directions. So after the ice retreated, one colonization route came from northeast, so basically through Russia and Finland, into northern parts of Scandinavia. And the other one came from southwest into southern parts of Scandinavia. And this is true for bears, and this is also true for many other organisms. And they kind of meet here in the middle of Scandinavia and overlap. And for bears, it's possible to trace this by looking at mitochondrial DNA, because these two different colonization routes carry a mitochondrial signature. And using reads from the dental calculus, we're able to reconstruct mitochondrial genomes of the bear specimens that we worked with, and show that those specimens collected in the north actually clustered with the eastern cluster that also contains Russian or Siberian brown bears, whereas those individuals collected in the south clustered with Western European cluster containing brown bears from Western Europe, and individuals collected in the middle actually belong to either one or the other. So this is the contact zone. So we can go beyond mitochondrial DNA, and we can also look at nuclear DNA. However, our ability there is quite limited because there are very, very few fragments of very little information within the dental calculus that corresponds to the nuclear genomes, but it is enough to ascertain at least the species. So we can show that our dental calculus samples here in kind of this bluish gray are clearly very distinct from polar bears, and they fall within the brown bear diversity, and more specifically, nuclearly, within the European cluster, or close to the European cluster. So we can do that. Right, and with that, I will jump into our first study that I wanted to present today, and this is a story of how we use dental calculus to track the history of human antibiotic use. And first, let's just recap what antibiotics and antimicrobial resistance is about. So antibiotics were discovered by Alexander Fleming in 1992 in a very, very, kind of strange way. Basically, he had contamination on one of his plates growing bacteria, and this contamination was a fungal colony. And he observed that all around the fungus, the bacteria were basically lice that were killed, and decided to investigate it further. And that led then eventually to the discovery of penicillin, the first antibiotic. And antibiotics started being mass produced, so basically on the industrial scale, in about 1940s. And it goes without any doubt and without any hesitation that antibiotics have saved millions and millions, if not billions, of lives. But they come at a price, and the price is antimicrobial resistance, which is rising worldwide. And current estimates say that this emerging resistance, being a global health threat, already causes about 700,000 deaths a year globally. And if nothing is done, the rising resistance will cause millions, the figure is about 10 millions of deaths, at about $100 trillion cost in fighting multi-drug resistance around the world. So it's a major, major health threat, one of the primary targets to fight against. So to understand this rising resistance, we also need to understand the antimicrobial resistance cycle, or AMR cycle. So basically, antibiotics, human-produced antibiotics, are being used primarily in two sectors, in the human biomedical sector and in agriculture. And both of these, so both human populations and animal populations, they obviously interact with each other, so domestic animals, and exchange antibiotics and potentially resistant bacteria. Through wastewater treatment, sewage, antibiotics and resistant bacteria get out into the wild environment, and from there they also get into wildlife. And of course, wildlife, domestic animals and humans interact with each other. So we have this very, very clear, network-like flow of resistant bacteria and antibiotics that are being exchanged between all these units. So if we want to understand how human use of antibiotics has impacted wild environments and wild animals, we could potentially study this. There are clear links. The problem is that antimicrobial resistance is actually a natural phenomenon. This is where we got our antibiotics in the first place. So if we want to understand the impact of humans on the resistance in the environment, we need to somehow subtract what is natural for this environment. It's not easy to do because antibiotics and resistant bacteria are spreading quite wide through wind and water processes. So resistant bacteria have been found in extremely pristine environments. So how do we characterize and quantify this natural antimicrobial resistance? And my take on that is that the only way to do this is to travel back in time to before humans started producing antibiotics. But before we do that, let me show you another slide about kind of a little bit the history of antibiotic use. Since we need to travel back in time, we need to know how far back we need to go. So before the 1940s, before the mass production of antibiotics started, this is a time period that we could call naive. So it's supposed not to be too heavily impacted by human antibiotic production. So for the first 20 years or so, between 1940s and 1960s, this is a period that's referred to as the golden age of discoveries. Different antibiotic classes were discovered. There was this major enthusiasm about using antibiotics. They have absolutely revolutionized medicine. But along with the discovery of each antibiotic class, resistances were also discovered. So this is not a new phenomenon. This has been observed quite soon after the first antibiotics were discovered. Nevertheless, for the next 30 to 40 years, there was a general, let's say, belief or general attitude that using more antibiotics or using antibiotics in combination would solve the problem of rising resistance. However, the only consequence of that was that resistance was on the rise. Until for the last 20 years or so, different countries and different continents and places worldwide have started to implement control measures to try and curb this rising antimicrobial resistance. And Sweden has been quite an interesting country in this respect, because Sweden was, in a sense, a little bit earlier than the majority of other countries. In particular, Sweden has introduced a ban on antibiotics in animal feed. So these are antibiotics that would be just supplemented into animal feed to increase growth. So they're basically growth promoters. So these were banned in Sweden in mid-1980s already. And in mid-1990s, Sweden introduced a strategic program against antimicrobial resistance, which is called STRAMA, which controls the sales of antibiotics in biomedical sector. And having lived in Sweden for 7 years, I can tell you from personal experience that it's almost impossible to get antibiotics prescribed unless everything else has been tried. So it's a very powerful and very strong program that encourages antibiotic stewardship. So we decided to see if this progress, so first of rising antibiotic use through time, and then the implementation of the control measures in Sweden specifically, if that had an impact on the levels and diversity of antimicrobial resistance that we can detect in wild environments. And for that, we turned to Swedish brown bears. And the question is, why would you ever do that? It doesn't seem like the most logical step. So let me walk you through our reasoning, and maybe it will make sense. So we decided to go for brown bears because they are widely distributed throughout Sweden, and they're omnivores. And there were a number of suggestions that omnivores might be quite good in refluxing antibiotics because they have this diverse diet, and maybe they will pick up antibiotics from multiple sources. A more kind of pragmatic reason was that we had access to an extensive collection of bear skulls from the museum that dated back to about 150, even 180 years. So it allowed us to cover this entire period of human antibiotic use, and to also the bears have dental calculus, obviously. And I can see there is a thing in the chat, which I cannot open. Okay, sorry. Great. Right. So basically, pragmatically, we're like, we have the correct collection. We have a species that builds up dental calculus. It makes sense to look at them. So let's do it. So this graph here will show you the levels of antimicrobial resistance in bear specimens going through time. And we will start with a naive time period. So each dot here is one bear specimen from which we collected dental calculus and quantified the proportion of resistance genes in this dental calculus with respect to all genes belonging to oral bacteria. And as we expected, we do detect antimicrobial resistance. Again, it is a natural feature of the environment, so it should be there. So then we sampled through time, going through this time period of big enthusiasm to time period of basically uncontrolled use and until presence. So we see an increase in antimicrobial resistance that corresponds to the increase in use of antibiotics through time in humans and agriculture. And what is quite interesting is that in the last 20 years, so since 2000, we see a decrease in the total AMR load, so in total antimicrobial resistance load, which corresponds to the time periods where the bears that we worked with lived their entire lives after the Swedish public health policy control measures were implemented. And this behavior of increase and decrease is significant. So it's a quadratic relationship between time and total AMR load. So what we also observe is that the diversity of resistance genes, here called families of resistance genes, actually has increased through time, which again corresponds to the diversity of different classes of antibiotics that are being used. And the reason for this increase is actually the appearance of low abundance new antibiotic classes. So sorry, I'll walk you through this. So we have antibiotic classes on the x-axis and we have time periods on the y-axis. There are certain classes that are always present, that change in abundance, but they're always there. And then we see an appearance in low abundance of novel classes in the last two time periods, corresponding to what we know in terms of use of antibiotics. So next things, we were interested to see if these levels of antimicrobial resistance are actually reflective of where we find our specimens. So here's again a map of Sweden, and the colored dots are the collection size of the bear specimens. And again, they have been collected through time, and underlaid in this gray, gray shade is the human footprint index. That is a measure of the impact of humans on the environment that takes into account the density of roads, the density of human populations, agricultural and industrial landscapes, and so on and so forth. So we wanted to know if bears that were collected close to human habitation or close to areas that are heavily impacted by humans would have higher antimicrobial load, because this is something that has been shown in other wild species. So first, we detected no effect of geography, latitude, longitude, municipality, or county. Because our sampling has been done through time, we actually looked at human population densities at the respective time when the bear has died. But we found no effect of historical human population density on antimicrobial resistance. And in fact, we found no effect of human activities taking the human footprint index into account. So none of the things that we expected to detect seem to have an effect. So we're wondering, why is that so? It has been shown for other species, so why don't we detect it in bears? And it seems that our bears are indeed wild animals. So here we have historical human population density. On the x-axis is the scale, so which values this population density can take within Sweden. And in blue, you see the Swedish average. And the vast majority, with only one exception of our bears, has been collected at population densities below the Swedish average. So our bears do avoid coming into human impacted landscapes. And the same goes for the human footprint index, for which we don't have historical values. So this is a value that has been calculated for 2009. But again, given the range and the Swedish average, the vast majority of our bear samples have been sampled from areas that are not heavily impacted by humans, which suggests that detecting the effects of increased antibiotic use and then the effects of implemented policies in the bears really suggests that the effect of antimicrobial contamination is widespread in the environment and is not only confined to the proximity of human habitation. So to conclude, human antibiotic use has a direct effect on the environment, on the levels of antimicrobial resistance and the diversity of resistant genes. And the environmental contamination with antimicrobial resistance factors, if you like, is really widespread and not limited to the proximity with humans. Right, and I'm looking at the time, and I will try to give you a little bit of a taste of a project we're currently working on. So it's very, very much work in progress. And maybe you have ideas, because that would be fantastic. So this is a project that is trying to understand how population demography can influence the health of wild animal populations through interactions with the oral microbiome. And the main idea hinges on this. So population declines may impact health through the interaction with the microbiome, and this is because the microbiome is controlled by these intrinsic host factors. So this is a principle that is based on microbiome as a system on a leash, where the host has a control over what grows and what is allowed to colonize the host and what doesn't. So if we kind of think this through, as genetic diversity of the host declines, as is happening for many wild animal populations today, so does the diversity of the host immune factors. This reduces the control of the host over host-associated microbial communities. And that, in fact, can then lead to the proliferation of pathogenic organisms and hence cause disease. So if that were true, we should see an increase in disease in those host populations that go through population declines, and we should be able to characterize pathogenic organisms in their microbiome. And again, this is where Swedish brown bears come handy, because they have experienced a severe population decline due to hunting. In particular, in about 1930s, the population was down to anywhere between 300 and 130 individuals. It has recovered today to about 3,000 individuals, but this 1930s population low seems to have been a true low. So we thought, great, fantastic, that's really great. So if we expect changes in the composition of the oral bacteria, then we have this temporal data set. Let's look into this. Maybe we see something. And we plotted one way and another way and looked at different things, and basically we found no difference in the composition of the oral microbiome in healthy bears that were sampled over the time period before, during, and after the bottleneck. However, what we did observe when we were working with museum collections was that bears have quite severe oral diseases. So here's a number of photographs showing this huge abscess here with like bone receding, so clear inflammation that has eaten away the bone, very, very clear cases of caries. So we're like, right, okay, do these communities differ at all? And in one of our first works, we have been actually able to show that if we sample from within such a cavity, here is the specimen, we can recover the presence of caries-associated bacteria here, showing the Streptococcus mutans, the classic for human caries, but also Lactobacillus casei and Paracasei, and it's only present in this diseased bear, but is absent in the healthy bears. Another just side note is that we were able to recover pathogens that are classically associated with human periodontitis, so rat complex bacteria. But in bears, we didn't observe any indication for health effects. So as suggested for humans, in fact, these bacteria seem to be normal members of the oral microbiome. Because we do shotgun metagenomics, we can go beyond just detecting the organisms and we can look at their functions. So we could show that this caries bear was enriched for functions that are related to sugar fermentation pathways and actually production of acids. And it was these bacteria that have significantly contributed at least to one of these functions, so to the remodeling of hexatol to lactate. Right, so we know we have oral diseases. We know these oral diseases have a clear pathogenic phenotype. We can quantify and characterize the species that contribute to them. So what we need to do now is to look at what happens to these bear populations through time. And I took advantage of two very enthusiastic students who didn't know what they actually signed up for and sent them into the museum to score skulls of bears for about a week. And there were quite a few skulls, and a few thousand, no, not a few thousand, a few hundred skulls. But so Maya and Jiansheng have been doing an amazing work. And based on their scoring of oral health, we could actually show that indeed, the proportion of diseased bears has changed through time in the Scandinavian bear population. And in particular, these are these blue bars because we need to account for uncertainty. And you can see that around the bottleneck here, sorry, the black line is the population of brown bears as far as we know it. And around the bottleneck, and following a few decades after the bottleneck, we see a high proportion of bear skulls showing presence of oral diseases. So as much as 50% of bears have been scored with having oral disease. And this proportion then declines as the population size increases. So indeed, we see that the oral disease is significantly more prevalent during the bottleneck and is less frequent in modern recovering populations. So looking at the microbiome, we can also clearly show that the microbiomes of the diseased bears are very, very distinct from those of healthy bears. And this is shown here. So the red dots correspond to bears with caries, and the blue and green dots correspond to either completely healthy bears or to bears that did have caries but were resampled from a healthy tooth. So these two are actually indistinguishable from each other, but the microbiome of diseased bears is very, very clearly distinct. And this disease microbiome is characterized by low diversity. So we look at alpha diversity, so diversity of microorganisms, and low evenness compared to the healthy microbiome, suggesting that it is dominated by few highly abundant taxa. So we decrease the diversity, but a number of those taxa that are still present become extremely abundant, and that's what decreases the evenness. So using this information, we of course can try and identify these putative pathogens and identify those that are most prevalent in bears. And surprisingly, we find exactly a number of pathogens that are associated with oral diseases in humans. And that of course allows us then to go further and say, okay, can we study the genomes of these pathogens in more detail? Can we also put them into evolutionary context of what we know about human pathogens? So that's what we did here. So this is Streptococcus mutans, one of the main drivers of human caries. And every single dot here is a human isolate of Streptococcus mutans, different human populations from all over the world. And this is where our bear sits. So it sits smack in the middle of the diversity that we know from humans. And we can also look at virulence and competence factors in this genome. And for instance, show that the bear pathogen here lacks a lot of the virulence factors that are frequently found in human Streptococcus. So we can start to understand the evolutionary history basically of pathogens with relevance to humans. So this is Streptococcus mutans. We can look at another pathogen that tells us a somewhat different story. This is Tenerella forsythia, one of the main bacteria implicated in periodontitis. And here the story is somewhat different because everything that we detect in our bears sits completely outside of the diversity seen in humans. So it depends on which pathogen we're looking at. So again, as I mentioned, this is very much work in progress, and we're very much in the beginning of this. But what I think we can see from these first set of analyses and thinking is that there seem to be a correspondence between prevalence of oral disease and population size in bears. What we're struggling with trying to understand is what is the cause? Because indeed, if our expectation is that declining populations and reduced genetic diversity is what enables the proliferation of pathogenic taxa, shouldn't we be seeing a much, much greater lag between population recovery and diseases, because population recovery will go slow in terms of increasing genetic diversity? So this is something that we need to still understand. We do have some ideas of how to go about this, but we haven't done it yet. We can clearly show that these disease microbial communities are dominated by few highly abundant putative pathogens. And we can study these pathogenic taxa and learn about their history and about their evolution. And in some cases, they fall inside the human diversity, in some cases on the outside. And this offers us the opportunities to study pathogen evolution. And I see Astrid comes online, and I'll be finishing quite soon. This is my concluding slide. I just want to demonstrate, or to summarize, that I believe that dental calculus is an amazing tool for ecology and evolution. It allows us to study oral microbiome evolution, dietary diversity, host evolutionary history, pathogens, antibiotics. So I think pretty much for me, this is can do it all tool, and I don't see why anybody wouldn't use it, but I'm quite biased. I would also like to acknowledge the museums that allowed us to work with their specimens and all the funding bodies. And the very, very last slide to finish is that we're continuing working on this. And I currently have a PhD position open that will continue working on uncovering the drivers of antimicrobial resistance in wildlife through time and space. And with that, I'm done. Super sorry that I went over time.